Eukaryotic cells. The eukaryotics include the protista, plantae, animal, and fungi. The eukaryotic cells, there are extensive compartmentalization of cytoplasm. Through the presence of membrane-bound organelles, eukaryotic cells possess of an organized nucleus with nuclear envelope. In addition to eukaryotic cell, have variety of complex locomotory and cytoskeletal structures. The genetic material are organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as form of possess of cell walls, plastids, and large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent almost in all plants. Then figure 8.3. A plant cell and B animal cell. What are the components of plant cell? Rough endoplasmic reticulum, smooth endoplasmic reticulum, lysosome, plasmodesmata, microtubule, perioxosome, cytoplasm, chloroplast, ribosomes, mitochondria, cell ball, middle lamella, vacuole, plasma membrane, nuclear envelope, Golgi apparatus, nucleolus, and nucleus. Then is animal cell. It have Golgi apparatus, smooth endoplasmic reticulum, nuclear envelope, nucleolus, nucleus, cytoplasm, rough endoplasmic reticulum, mitochondria, ribosome, lysosome, perioxosome, centriole, plasma membrane, microvilli. Then cell membrane. The detailed structure of membrane was studied only after the advent of the electron microscope in the 1950. Meanwhile, chemical studies on the cell membrane, especially in human red blood cell, enabled the scientists to deduct the possible structure of plasma membrane. These studies showed that cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in bilayer. Also, lipids are arranged within the membrane with the polar head towards outer side and the hydrophobia tails towards the inner side. This ensured that non-polar tail of saturated hydrocarbon is protected from aqueous environment. In addition to phospholipid, membrane also contains cholesterol. Later biochemical investigation clears that reveals cell membrane also possess protein and carbohydrate. The ratio of protein lipid considered in different cells. Then human being, the membrane of erythrocyte has approximately 52% protein and 40% lipid depending upon ease of extraction. Membrane protein can be classified as integral and peripheral. Peripheral protein lie in the surface of membrane while the integral protein are partially or fully buried into the membrane. Then an improved model of structure of cell membrane was proposed by Singer and Nicholson in 1972. Widely accepted as Fluid's mosaic model. According to these, the fluid nature of lipid enabled lateral movement of protein within the overall bilayer. The ability to move within the membrane is measured as fluidity. The fluid nature of the membrane is also important for the point of view of junction like cell growth, formation of intercellular junction, secretion, endocytosis, cell division. One of the most important functions of plasma membrane is to transport of molecule across it. The membrane is selectively permeable to some of the molecule present on either side of it. Many molecules can be brief across the membrane without any requirement of cell, and this is called passive transport. Neutral solutes may across the membrane by the process of simple diffusion along concentration gradient from higher concentration to lower. Water may also move across the membrane from higher concentration to lower concentration. Movement of water by diffusion is called osmosis. As polar molecule cannot pass through the non-polar bilipid layer, the required carrier protein of the membrane facilitates the transport across the membrane of few ions or molecule are transported across the membrane. Again, the concentration gradient from lower to higher concentration. Such a transport energy dependent process in which ATP utilized is called active transportation. Example: sodium potassium pump. Cell wall. As you may recall, a non-living rigid structure called cell wall formed an outer layer covering from the plasma membrane of fungi and plants. Cell wall not only given shape to cell and protect cell from mechanic damage and infection, it also help in cell to cell interaction and provides barrier undesirable macromolecules. Algae have cell wall made of cellulose, lactase, mannose, and minerals like calcium carbonate, while other plant consist cellulose, hemicellulose, pectins, proteins. The cell wall of young plant is primary wall, the capable of growth which gradually diminished as cell mature, and the secondary wall formed on inner side of the cell. The middle lamella layer of calcium pectate which holds glues of different neighboring together. The cell wall of middle lamella may be transversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While the membranous organelles is distinct in terms of its structure and functions, many of these are considered together as endomembrane system because their function and coordination. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Since the function of mitochondria, chloroplast, and peroxisome are not coordinated with the above components, these are not considered as part of endomembrane system. Endoplasmic reticulum. Electron microscope studied in eukaryotic cells reveal the presence of network of reticulum of tiny tubular structures scattered in cytoplasm that is called endoplasmic reticulum. Hence, ER divides intracellular space into two distinct compartments, luminal and extraluminal. Luminal is inside endoplasmic reticulum and extraluminal is cytoplasm compartment. The endoplasmic reticulum often shows ribosome attached to their outer surface. The endoplasmic reticulum bearing ribosome on the surface is called rough endoplasmic reticulum, and the is absence ribosome, they appear as smooth and they are smooth endoplasmic reticulum. RER is frequently observed in cells actively involved in protein synthesis and secretion. They are extensive, continuous, and outer membrane nucleus. The smooth endoplasmic reticulum is major site of synthesis of lipid in cell. Lipid-like steroidal hormone synthesized in SCR. Golgi apparatus. Camillo Golgi, 1898, first observed densely strained reticulum structure near nucleus. These were later named as Golgi body after him. They consist of many flat disc-like sacs or cisternae of 0.5 nanometer to 1 nanometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distant convex cysts of forming face and concave trans or maturing face. The cis and trans face of organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to intracellular targets or secretes outer the cell. Material to be packed in form of vesicle from ER fuse with the cis face of Golgi apparatus. The moment of maturing phase, this explained by Golgi apparatus, remains in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosome on endoplasmic reticulum are modified by cisternae of Golgi apparatus before the release from transphase. Golgi apparatus is the important site of formation of glycoprotein and glycolipids. Lysosome. These are membrane-bound vesicle structure formed in the process of packaging of Golgi apparatus. The isolated lysosomal vesicles have been found in very rich in amount of all these hydrolytic enzymes. Hydrolyses are lipase, protease, carbohydrates optimally active at all pH. The enzymes are capable of digesting carbohydrate, protein, lipids, and nucleic acid. Vacuoles. A vacuole is the membrane-bound space found in cytoplasm. It contains water sap, secretory products, and other material not useful for the cell. The vacuole is bound by single membrane called tonoplast. In plant cell, vacuole are occupied 90% of volume of cell. In plant, tonoplast facilitate transportation of number of ions in other material against the concentration and gradient into vacuole. Hence, their concentration significantly higher in vacuole than the cytoplasm and ammonia. The contractile vacuole is important osmoregulatory and secretory in many cells as a protista. Food vacuole are formed engulfing the food particles. Mitochondria. Mitochondria, unless specially stained, are not easily visible under microscope. The number of mitochondria per cell is variable depending on physiological activities of the cell. In terms of shape and size, considered degree of variation is observed. Typically, it is sausage shaped or cylindrical, have diameter 0.2 to 1 micrometer, and length 1 to 4 micrometer. Each mitochondria is double membrane wound structure with the outer membrane and inner membrane divide its lumella distantly into two aqueous compartments, the outer compartment, inner compartment. The inner compartment filled with dense homologous substance called the matrix. Outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfolding called cistrae towards the matrix. The cistrae increase area, surface area. Two membrane have their own specific enzyme associated with mitochondrial function. Mitochondria are site of aerobic respiration. They produce cellular energy to form ATP. Hence, they are called powerhouse of cell. The matrix of process single circular DNA, few RNA, ribosome 70S are the component required to synthesize of protein. The mitochondria